Friends, we are back again with our 8th tutorial video, that is on fluorescent microscope. This microscope I am calling it as fluorescent microscope, but it is more than fluorescent microscope. It has two other more functions. One is bright field and another is dark field. Now what is this bright field microscope? Bright field microscope is just our normal microscope. It is a normal slide microscope. This is basically a slide microscope. Actually, the bright field option is that it can when the periphery of the when the light passes through the object, the periphery becomes lighted, illuminated. The whole periphery is illuminated, so that is called bright field. The field is bright, so field is illuminated, so the field is bright. And in the dark field, what happens that when the periphery, there's an adapter, it's a dark field condenser is there, through which the light passes, but it allows only the light to pass through the object or the sample or the specimen. It should pass through the specimen, and the other periphery is dark, so we can see only the object and not the periphery. So that is very it makes us more convenient to see the object properly, or to view the specimen properly. So that is dark field microscopy. Now what is fluorescence microscopy? Fluorescence is a very beautiful phenomenon. It is an optical phenomenon. It is a chemo-optical phenomenon rather. What happens so that when a specimen or a sample is stained in a special type of stain, it does not show any emission in the bright field or in the dark field. It starts showing the emission in the presence of ultraviolet light. When an ultraviolet light of a certain frequency of 400 nanometer, 400 to 430 nanometer is placed, or it falls on the object slide, a fluorescence is emitted. A second color is emitted. That emitted color is different, or in the lower wavelength, and some higher frequency color is focused on that object. The lower frequency is emitted. So this is called fluorescence, and this fluorescence is present. It takes place in the presence of that ultraviolet light. When that ultraviolet light is switched off, then there is no fluorescence. When there is a presence of ultraviolet light, this fluorescence emission starts, and this helps us to detect more phenomena which are not visible in the bright field or in the dark field. So what is those things? The main thing is the mycotoxin present in the fungus, the DNAs, or the presence of cell proteins or cell nucleus and all that gets visible in this fluorescence microscopy. And so that is a very beautiful microscope and very beautiful phenomenon by which we can detect the unknown or invisible things which are invisible in the bright field and the dark field microscopy. It regards a very little bit lengthy process of processing the specimen, and then it regards the staining process. This staining requires two stains. One is rhodamine auramine, and other one is IRCP stain. So indigo caramine, rhodamine auramine confocal stain. So that is a different stain. So these two stains are mainly incorporated. So that we will see in any other video. So now we are consisting of this fluorescence microscope. Now how this fluorescence microscope works and what is the objectives of the fluorescence microscope, how it looks like. So that we will see in the next part of the video. So that is also a LABOMED EFL fluorescence microscope. Now what is this EFL? That we will also see. What is EFL? So that we will see. So that we will be recording in the next part of the video. So be with us to see this tutorial and see this LABOMED EFL, LABOMED LX400 EFL microscope. This is an upright microscope, not like that inverted tissue culture microscope. This is an upright microscope, so you will be able to see all the details. Perhaps I will be able to show you perhaps all the details of this microscope, how to use it and how to see the slides and all that we will see in the next part of the video. So be with us. Hello friends, so this is our bright field, dark field and fluorescent microscope. So this is the upright microscope, as you can see, the objective. These are the objectives. So these are the objectives, you can see. So these are there are 4 objectives are there, and this is our eyepiece or the binocular in the top. This is our binocular, and this is the triangular head. And if I little bit move the camera, you will be able to see the triangular head. So this is the triangular head, and above you can see the camera. So now we are going to see the parts of this microscope. So one thing you can see, an extra thing in the side of that is the power supply. So this is the power supply, and this is the power on switch. So I am switching on the power, and the microscope is powered on. And there is a back, at the back there is another switch which changes from bright field. So this is bright field mode, and this is switched off. The microscope is switched off, and this is the UV mode. So UV, for that UV mode, you have to put this. There is a plunger. You have to pull this plunger in this side of the microscope, which I will show you. So this is the plunger. This is the plunger is switched off. The UV light is off. Now the UV light is falling. So this is the dark field. Now this is the dark field, and you can see that the UV light is there on the slide, and you will be able to see through this different eyepiece, through these different eyepiece. So this is our mainly the UV, the bright field and the dark field, bright field, dark field, and the UV microscope or the fluorescence microscope. Now here is the slide. This is the slide, and this is the stage. You can bring the camera here. So if I bring the camera a little bit nearer, you can see the stage. So here is the stage, and this is the stage, and you can place the slide over here. So this is the slide. So here you will be able to see through this binocular eyepiece. Little bit camera if I move, and you can see the binocular eyepiece. So this is our binocular eyepiece, and you can able to see through as the same as this. And this middle part here is the fluorescence part, which you can see little bit from this side. If I bring this side, the camera, yeah, this is the fluorescent attachment. This is the fluorescent attachment, and this is the, and if you can bring the camera a little bit, if I move the camera a little bit, and the camera is blocking, but you can see if I move the camera from this side, if I turn the camera, so just I am turning the camera, and you will be able to see the other part of the fluorescent microscope, the other side of the fluorescent microscope. This plunger is the shutter of the UV mode. So this closes the UV light, and this opens the UV light. This switch on the UV light. So the UV light passes through this condenser, this EFL. From this EFL adapter, it passes down to the objective, and it falls on the eyepiece. So it falls on the slide. So here you can see. Now if I move this cover, you will be able to see the EFL light. Yes, you can see the EFL light. So from this UV adapter, the UV light is coming down through these lenses, these UV lenses, and it is falling on the slide. And this light is also going up, and it is going to the objective. This objective, you can see these objectives, and this objective is little bit forward, and this objective, and you can see through this. So this is the UV system, and as you can see from this side also, you can see there is a put down the camera, and put down the camera. You will see the controls over here, and little bit that side you can see the control, and this is the control for, you can see from this side, this is the control. This is the control from this side. You can see this is the control, and this control actually decreases or increases the UV light. So this is the UV light. Intensity of the UV light is decreased, and if it turn this towards you, the UV light is increased. So this how the EFL works, and this closes the UV light, and this increases or decreases the UV light. And here is the adapter, and you can take the picture. So this is the fluorescence part, and now this condenser, you can see this condenser is showing this condenser. This is our dark field condenser. So this goes inside under this UV light, under this stage there is a condenser. If I put down the camera a little bit, if I put down the camera a little bit, and you can see the condenser below. The stage, there is a condenser. This is the condenser. So this condenser, this is a bright field condenser and UV condenser. So this is taken down. This condenser is taken down. You can, there is a knob. In this side there is a knob, and the condenser is coming down. So you can see the condenser now. Now you'll be able to see the condenser. So if I put some little bit external light from here, we'll be able to see the condenser. Now here you can see the condenser. So if I put some external light, so you can see the condenser. So this is the bright field control. This is the dark field mode, and this is the bright field mode. So this you can control. From here, this lever controls the dark field and the bright field mode. This lever controls the dark field and the bright field mode. So this condenser can be removed with the help of screws are there. So this can be removed and we can place the dark field condenser over there. So then it will become the dark field microscope. Now this bright field and UV setup is there. So if this is removed, then the dark field condenser can be used. So this is our dark field condenser. So that is it. So this is our bright field, dark field, and UV light, UV microscope. So it is up to you that how you use it. So here one thing you should be careful about the slide, the placement of the slide. One thing you should be careful about placement of the slide. Now this is the slide, you can see. So this has a cover slip on it. So the cover slip side should be upwards. It is like this. It should not be like this. So it should be upwards to focus the light properly. So you can see the slide, and this is the slide. So this is all about the fluorescence, dark field, bright field, and the fluorescence microscopy. So thank you very much.